So let's talk about blood vessels now. Earlier I said that blood vessels act as a channel to guide blood to different parts of our body. Largely speaking, there are three groups of blood vessels. You have blood vessels which carry blood away from the heart, and we call them arteries. Then you have blood vessels which carry blood back to the heart or toward the heart. We call them veins. And then there are small microscopic blood vessels which connect artery and vein, and those are called capillaries. Capillaries are also actually the site of exchange for oxygen and nutrients. I also want to mention that smaller arteries are called arterioles and smaller veins are called venules. Now let's look at the structure of a blood vessel. In this slide you see two types of blood vessel. You see an artery on the left and a vein on the right side. So these arteries and veins actually indicates large blood vessels, because in capillaries or in smaller blood vessels you may see a thinner layer of blood vessels. So large blood vessels are made of three layers. Regardless if it's an artery or vein, they're made of three layers. So from inside to outside, these layers are tunica interna, tunica media, and tunica externa. Tunica interna is made of simple squamous epithelial cells. They make up a smooth surface for the blood so they could easily flow. The difference between artery and vein is that the tunica interna of vein, you also find these flap-like structures which serve as a valve. So veins, medium to large veins, they may have valves as well, while arteries do not have valve. The next we have tunica media, which is made of smooth muscle. The difference between artery and vein is tunica media is slightly thicker in artery. The smooth muscle actually causes constriction, and that determines the diameter of the blood vessel, which acts which influence blood flow and blood pressure. And then finally we have tunica externa, which is made of connective tissue. It's similar in both arteries and veins. The tunica externa not only protect the blood vessels, but it also anchors or attaches the blood vessel to the surrounding organs. I also want to add that although arteries are slightly thicker than the veins, but veins have larger diameter compared to the artery. So for this reason, veins are also called capacitance blood vessels, because they carry most of the blood. They contain or they have most of the blood in it. This slide shows the structure of a capillary. So capillaries are microscopic hair-like structures. Compared to a large blood vessel, they're only made of one layer of cells. The type of cell is actually simple squamous epithelial cells. But the unique thing about, or the unique point about capillary is that there's a gap between the cells, and that allow the capillary to serve as an exchange site for gases, for nutrients, and other substances. So as I said, that exchange occur at the level of capillaries, and by exchange I mean that some substances move from inside of the blood vessel to outside of the blood vessel, and this is called filtration. And on the other hand, you have some substances that move from outside of the blood vessel to inside of the blood vessel, and that's called absorption. There are forces that facilitate absorption or filtration. So for example, at the arterial end of the capillary, the blood pressure facilitates filtration, and it causes the substances to move from inside of the blood vessel to outside of the blood vessel. Compared to the venous end of the capillary, where you have more of absorption, and the reason for that is that there's a relatively less blood pressure. However, there's another type of pressure as well, and that facilitates absorption, and that's called colloid osmotic pressure. The colloid osmotic pressure is generated by protein, especially albumin, and it attracts water. So if you do the math on the arterial end of the capillary, there is more outward pressure than inward pressure. So the overall net result is that fluid will move outside, and that is basically filtration. However, at the venous end of a capillary, the outward pressure is less than the inward pressure. So overall, the inward pressure wins, and that's why fluid moves inside the cell, and that makes up the absorption. I briefly mentioned earlier about blood pressure, that it facilitates filtration. So now let's discuss what is blood pressure. So blood pressure is actually the pressure which is exerted by the blood on the wall of blood vessel. And we usually refer to the blood pressure as the arterial pressure. There is some pressure in the veins as well, but we primarily focus on the pressure in the arteries. This slide shows the tool which we use in order to measure blood pressure. This tool is called sphygmomanometer. Sphygmomanometer is made of an inflatable cuff which we wrap around the patient arm. It has a bulb or a balloon-shaped structure which we use in order to build up a pressure in the cuff. It has a pressure gauge which we use in order to read the pressure inside the cuff, and we also use a stethoscope in order to hear the sound. This is an important graph. Let's pay attention here. So on the x-axis we have the distance from the heart, and on the y-axis we have pressure. We have pressure in the arteries. So as you can see that as we move away from the heart, the pressure actually drops down. Okay. Also, you need to know that there are two types of pressure. We have systolic blood pressure and diastolic blood pressure, and that refers to the activity of the heart. When heart contracts, obviously the pressure builds up, and that is called the systolic pressure. And when the heart relaxes, the pressure goes down, and that is called diastolic pressure. Pay attention that when the heart relaxes, the pressure does not go down all the way to zero, and that is because the tonicity or the contraction of smooth muscle in the blood vessel actually generates some pressure which is responsible for diastolic blood pressure. But as we move away from the heart, it's hard to differentiate between systolic and diastolic pressure, and as you can see, when we get to the veins, especially large veins, the pressure gets to almost 5 to 10 millimeter mercury. There are several factors which can influence blood pressure. For example, cardiac output, blood volume, peripheral resistance, and blood viscosity. First, let's talk about cardiac output. So what is cardiac output? I want to first clarify another term, and then I'll get back to cardiac output. So that term is stroke volume. Stroke volume is the amount of blood which is pumped out of the heart with each contraction. Okay, not per minute, per contraction. So if we calculate this per minute, that will give us cardiac output. Now I can define cardiac output. Cardiac output is the amount of blood which is pumped out of the heart during each minute. I think students confuse between cardiac output and stroke volume. Just remember that stroke volume is per beat, amount of fluid pumped out of the heart per beat or per contraction, while cardiac output is amount of blood pumped out of the heart per minute. There's a formula you can use to measure cardiac output, and that's stroke volume times heart rate, because we calculate or measure heart rate per minute. So we can, instead of using 1 minute, we can just put the numbers of heart contraction per minute. So the normal stroke volume is around 70 milliliter, and the normal heart rate is around 70 to 75 beats per minute. So if you do the math, the cardiac output is almost equal to 5 liter, and that's the entire blood. So our heart pumps the entire blood each minute. This slide shows various factors that affect blood pressure. So for example, when blood volume goes up, pressure goes up. When blood volume goes down, pressure goes down. Same applies to the heart rate and stroke volume. Also, think of a case in which our blood become more thicker. For example, in the case of spherocytosis, there are more red blood cells. So it makes the heart job a little bit harder in order to pump thicker blood. That's why the pressure goes up. But probably the most important factor that determines blood pressure is peripheral resistance, and especially the blood vessel diameter. So if the blood vessel diameter is increased, that causes the blood pressure to drop down. And when the blood vessel diameter is shortened because of blood vessel contraction, that increases the blood pressure. A lot of the cases when blood vessel dilates, this leads to low blood supply to the brain, which can lead to unconsciousness and even coma and death. So in different part of the cardiovascular system, and specifically in aorta and in carotid artery, there are receptors which sense any change in blood pressure. Those receptors are called baroreceptors. So whenever there is a change, baroreceptors will sense that change, and then they will carry the information, send the information to the cardiovascular system in medulla oblongata, which is part of the brain. And then in response to that, the medulla oblongata, or the cardiovascular system, cardiovascular center, will activate the autonomic nervous system, which includes sympathetic and parasympathetic nervous system. So think of a case in which blood pressure is low. That will be sensed by baroreceptors, and then the information will send to the cardiovascular control center, and the cardiovascular control center in this case will activate the sympathetic division, which is also called cardio accelerator system. This will increase the blood pressure by squeezing the blood vessels, which is called vasospasm, and also increase the heart ability to contract faster and stronger. That basically means increased cardiac output. You can also think of the other way around, or the opposite case. For example, somebody's blood pressure is high. That will be sensed by baroreceptors, and the information will be sent to the cardiovascular control center in medulla and medulla oblongata. This time activates the parasympathetic nervous system, and that is basically the cardio inhibitor branch, and that will lead to relaxation of blood vessel and also relaxation of the heart, and as a result blood pressure will decrease. The last point that I want to add to this lecture is that in the arteries, blood flow due to the pressure generated by the heart. How does the blood flow in the veins when there is almost no pressure? So then there are at least 3 mechanisms which helps the blood flow in the venous system in order to return back to the heart. One is the contraction of the skeletal muscle. So when we use our skeletal muscle, that squeezes the blood in the veins and helps it to move forward in the venous system. Then we have the respiratory movement. So every time we inhale, our lungs act as a suction, and it helps to bring the venous blood back to the heart. And finally, venoconstriction. So there is a thin layer of smooth muscle in the wall of vein that also help to a certain extent in order to squeeze the blood and send it back to the heart. And this concludes our lecture on cardiovascular system.